Hello everyone, thanks for coming today. And today I will talk about decoding genomic patterns involved in sepsis using data-driven machine learning approaches. Data from multiple omics technologies is very common nowadays. Different blocks of omics data are shown in blue here. For example, there could be a proteome, metabolome, and transcriptome. Features are shown in orange, the individual biomolecules associated with each omics data. For example, there could be individual transcript, proteins, or metabolites. Our main motivation for investigating data from a multi-omics angle is that we hypothesize that when a biological system is perturbed, there are changes at every level of omics data, and these changes need to be analyzed together to get an integrated view of the system and to understand the correlation between involved biological molecules shown by these arrows here. We have some empirical evidence showing that multi-omics analysis are consistently more accurate than single omics analysis. I show the minimum error rates obtained for each analysis on the COVID-19 case study on the left. The orange box plots show the error rate distribution for single omics analysis, and the overlaid blue line shows the minimum error rate obtained for multi-omics analysis on the same dataset. And you can see that multi-omics approach consistently outperforms the single omics approaches. Having demonstrated that multi-omics provides a higher resolution view of data, we then consider that it's not trivial to analyze multi-omics data, as the variety of data modalities and formats are very large. To address this barrier, we created a multi-omics data integration framework to investigate bacteria that cause sepsis in hospitals. And to do this, we use a combination of machine learning and natural language processing. We apply this to investigate the functional and regulatory signatures of sepsis and to identify integrated features of interest at the molecular resolution. In many cases, these microbes are resistant to at least one antibiotic. And to design targeted drugs of sepsis, we need to develop biomarkers and characterize the gene regulatory pathways of these microbes. So we use a prokaryotic dataset from an Australian national framework project on antibiotic-resistant bacteria. We have 3 omics data, including the genome, and the conditions in this study is the growth media, which is RPMI and pooled human sera. We constructed a pipeline which is able to perform this multi-omics data integration. It is distinct from conventional multi-omics analysis, which aggregates summarized data into pathways. And in our case, we identify the correlations between individual biological molecules in each omics data. And on the left is a single omics data analyzed as part of this consortium. This is an upset plot of pathways. You can think of this as a quantitative Venn diagram. On the right, we can find the correlation between the individual biological molecules in each block of omics data. On the left, the highest resolution is the pathway. This again is another supporting point for the power of multi-omics data analysis. So the conventional aggregative approach implicates histidine, glucose uptake, and TCA cycle pathways as involved in sepsis. And this we also find some on the right, we have the single molecule resolution version, which detects the specific genes HISB, SERT, and SA, which are involved in these pathways. In addition, we also detect some proteins and metabolites. Not all detected features are annotated, and some are unknown, and our pipeline is able to detect these. From this, we analyzed the intergenic regions of an unannotated gene in the previously newly sequenced bacterial strain. We discovered various patterns of fixed length, which we investigated further. We can identify short motifs such as nucleoid protein binding domains, and we're able to recapitulate long motifs as a composite of several short motifs. In this case, we identified the cis regulatory region. So sequences we had no known annotations also discovered, and this applies to unknown unannotated genes as well. Here we identified a sequence in a newly sequenced genome corresponding to a motif regulating deactivated Shigella toxin, which a frameshift can reactivate. Since it does not rely on a motif database and is purely data-driven, discovering novel motifs is also possible, as I mentioned. So we're able to detect highly correlated functional omics, genomic, and regulatory features in sepsis. This method can accept raw sequence data with no prior annotation. Overlaying this genomic and regulatory features on functional omics data allows us to identify interconnections between the biomolecules responsible for driving change in the disease system, which will lead to a better understanding of the molecular mechanism of sepsis-causing bacteria. 27 bacterial strains are available, and we will run a pipeline on these, refining it further when needed. Thank you all for listening.